Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway and cancer. Okay, right. So in the previous video what we did is we went over the basic carcinogenesis process, and we saw that an absolutely key property of cancerous cells is that they over-proliferate. In fact, this is believed to be the first change that occurs to that somatic cell, which starts it on the progression towards becoming cancerous. Okay, we then discussed the involvement of the PI3 kinase AKT mTOR pathway in causing a cell to divide. Okay, we discussed that when a cell takes this decision to divide, firstly it has to grow and then it will split into two. Okay, and this requires a major epigenetic shift. Basically, you have to start expressing a whole bunch of genes that you weren't expressing before and upregulate the expression of a whole bunch of genes that you might have been expressing before. So it causes a major change in the transcriptome and the proteome of this cell, which just means the collection of all mRNAs of a cell, that's the transcriptome, and the collection of all proteins in the cell, that's the proteome. Okay? Right, and we discussed that the way that you produce this epigenetic shift is twofold. Okay, firstly you have to produce the transcriptional change. You have to now transcribe the mRNAs. Okay, you have to transcribe new genes that weren't being transcribed before, or increase the transcription of genes that were being transcribed before that need to have their transcription altered. Okay, and this change in transcription within the cell is produced by pathways such as the RAS, RAF, MEK, ERK pathway, okay, which change the transcription. Okay, they alter epigenetic control via transcriptional control. However, the PI3 kinase AKT mTOR pathway, that is involved in translational control. Okay, so if you just had the transcriptional control and just made all of these new mRNAs as a pro-division, okay, it doesn't mean anything. They have to now be translated into proteins for them to actually have an effect. Okay, and basically there's normally a blockade on mRNAs that are pro-division being translated, as we'll discuss later on. They are what are known as elongation factor 4E sensitive mRNAs. Okay, and there's usually a block on the translation of these sorts of mRNAs. Okay, so if you now want them to actually be translated and actually have an effect on the proteome of the cell, you need to allow them to be translated, basically, and that's done by the PI3 kinase AKT mTOR pathway. It releases the brake on the translation of these pro-division mRNAs. Okay, right. So we now want to discuss this PI3 kinase AKT mTOR pathway, and we're going to begin by discussing PI3 kinase, phosphoinositide 3 kinase. So let me just explain how I'm going to structure our discussion of this pathway in depth. We're going to start with PI3 kinase, then we will go backwards and look at how PI3 kinase can be activated. Okay, so we're going to look at the enzyme before we look at how it can be activated. Okay, and you might ask, well, why are you not doing its activation before we discuss its function? Well, because there are many different ways that PI3 kinase can be activated. Okay, it can be activated by receptor tyrosine kinases, it can be activated by G protein coupled receptors, it can also be activated by small GTPases. Okay, so we're going to discuss the enzyme first and then go back to its activation. Okay, and then we'll go on with the pathway. We'll go on to AKT and then mTOR. Okay, right, so let's start by discussing phosphoinositide 3 kinase then. So that's what PI3 kinase firstly stands for. So PI3K, which we've been writing for so long, stands for, the PI is for phospho, that's the P, inositide, that's the I, and then it's 3 kinase. Okay, so phosphoinositide 3 kinase enzymes are enzymes which phosphorylate lipid molecules. They phosphorylate phosphoinositides. Okay, specifically they phosphorylate phosphoinositides on the third carbon of the inositol ring, as we'll see in a moment. Okay, right. Now, there are three broad types of phosphoinositide 3 kinases, which are known as type 1 phosphoinositide 3 kinases, type 2 phosphoinositide 3 kinases, and type 3 phosphoinositide 3 kinases. The ones that we are interested in, the ones that most is known about, are the type 1 phosphoinositide 3 kinases. Okay, so we're not going to discuss in this video at all the type 2 or the type 3 phosphoinositide 3 kinases. Okay, the three different types of phosphoinositide 3 kinases, they add phosphate groups onto the same position of phosphoinositides, but the phosphoinositides that they target are very different. Okay, so their substrates are different. Type 1 phosphoinositide 3 kinases, their substrates are, their substrate is phosphatidyl inositol 4,5-bisphosphate, which is often abbreviated as PIP2, okay? And this is the important one in this pathway that controls the translation of these elongation factor 4E sensitive mRNAs, okay, which are pro-division mRNAs generally. Okay, so we're only going to concentrate on type 1 phosphoinositide 3 kinases, which have as their substrate PIP2. Okay, so let's now talk about PIP2 then, because this is going to be very, very important. So PIP2 stands for phosphatidyl, okay, that's the P, then inositol, that's the I, and then it's phosphatidyl inositol 4,5-bisphosphate. So the second P in the PIP2 is for phosphate, and then because you've got two of these phosphates added onto the phosphatidyl inositol, it's P2. Okay, now, if you're being slightly more strict, you would abbreviate this down not to PIP2 like so, but instead you would abbreviate it down to PI45P2, okay, because this abbreviation here doesn't actually tell you which carbons of the inositol ring the two phosphate groups come off, whereas this abbreviation does tell you that they come off the 4th and the 5th. Okay, now, the most important example of a phosphoinositide with two phosphate groups coming off the inositol ring is this one where they come off the 4th and the 5th carbons, and therefore if you wrote PIP2, people would assume that you meant phosphatidyl inositol 4,5-bisphosphate rather than maybe phosphatidyl inositol 3,4-bisphosphate. Okay, but if you want to be strictly correct, you'll see it abbreviated down to PI45P2. Okay, right. So what I now want to do is discuss this molecule. Okay, so it is a normal component of the phospholipid bilayer of the plasma membrane. Okay, it's quite a rare phospholipid to find in the plasma membrane. Okay, but it is there, is the important thing. So you have it in the cell membranes of all of your cells, basically. Okay, so let's discuss the structure of phosphatidyl inositol 4,5-bisphosphate. Now, we're not going to draw out an actual chemical formula for it. What we're going to do is draw out a cartoon for what it looks like so that we can understand what phosphoinositide 3 kinase type 1 enzymes are going to do to it. Okay, right, so let's draw the plasma membrane here. Okay, and this line here is going to represent the outer leaflet of the plasma membrane, and this line here is going to represent the inner leaflet of the plasma membrane. Okay, now, phosphatidyl inositol 4,5-bisphosphate is going to be a component, it's going to be a phospholipid within the inner leaflet of the phospholipid bilayer, so let me show it here. Okay, so we'll start off, we'll go through its name in parts, okay? So we'll start off with the phosphatidyl. Let's try and understand what this phosphatidyl prefix that we have in the name means here. Okay, so what this means is that the molecule is based on another molecule called phosphatidic acid. Okay, so phosphatidyl inositol 4,5-bisphosphate is going to contain phosphatidic acid. Okay, so what's the structure of phosphatidic acid? So phosphatidic acid is based on glycerol, okay? Glycerol is the backbone for phosphatidic acid. So this horizontal line that I've now coloured in green, this is going to represent a glycerol molecule, a 3-carbon molecule with alcohol groups coming off each of the 3 carbons. Okay, so remember the more correct name now for glycerol is propane 1,2,3 triol. Glycerol is the old biochemist's name for this this molecule, the new chemistry name is propane 1,2,3 trial, which tells us that it is a 3-carbon molecule, propane, where you've got alcohol groups coming off the first, the second, and the third carbons. Okay, right, then what you have done is attached onto the alcohol group coming off the first and the alcohol group coming off the second carbons. You've attached on long-chain carboxylic acids, which are these vertical lines in orange here. Okay, so these are representing fatty acids. Now, a fatty acid is just a carboxylic acid which has a really long hydrophobic tail. Okay, so these are the lipid molecules that you have got in this molecule, basically, and these are what are holding it attached to the phospholipid bilayer. Okay, so long-chain carboxylic acids, and they've been esterified onto the alcohol groups of the first and second carbons of the glycerol molecule. Okay, right. Then the final thing that makes phosphatidic acid is that you then have a phosphate group, which is this purple blob here, which is attached onto the alcohol group that comes off the third carbon of the glycerol molecule. Okay, so this is a phosphate group. And that now is the complete phosphatidic acid molecule, and hopefully you'll agree that this is an example of a phospholipid, okay? Because the characteristics that you need in order to be a phospholipid molecule are you need to have a phosphate group, that's one of the characteristics, and the other characteristics you need are a lipid component. Okay, and we've got 2 lipid components here, these 2 long-chain carboxylic acids, esterified onto the alcohol groups of the first and second carbons of the glycerol molecule. Those are our lipid components. Okay, so phosphatidic acid itself is a lipid molecule. So this is my cartoon, it's a phospholipid molecule. So this is my cartoon for phosphatidic acid here sitting in the plasma membrane. Now, we want to convert phosphatidic acid into phosphatidyl inositol 4,5-bisphosphate. So the first thing we'll do is convert it into phosphatidyl inositol. Okay, so to convert phosphatidic acid into phosphatidyl inositol, what you need to do is attach onto this phosphate group here an inositol ring. Okay, now an inositol ring is a 6-carbon ring, okay, where you have alcohol groups coming off all of the 6 carbons. So the correct name for inositol is cyclohexane 1, 2, 3, 4, 5, 6 hexol. So 1, 2, 3, 4, 5, 6, and then hexol. Okay, right. So basically it's a 6-membered carbon ring where the bonds between the 6 carbons are single bonds, and you've got alcohol groups coming off the first, the second, the third, the fourth, and the 5th and the 6th carbon. So every carbon has a single alcohol group coming off, and then to make up the final bond, a single hydrogen coming off. Okay, now you're going to attach the alcohol group of this carbon here, which is the first carbon, to the phosphate group here via a phosphoester link. Okay, and that's how we've attached the inositol molecule onto the phosphatidic acid molecule. This molecule that we've now created here is what's called phosphatidyl inositol. Okay, now if we want to turn it into phosphatidyl inositol 4,5-bisphosphate, we have to stick 2 more phosphate groups off the inositol ring. And specifically, we need to put them off the 4th carbon's alcohol group and the 5th carbon's alcohol group. Okay, so this is the first carbon here. Now we'll just count round 2, 3, 4, 5. So we want them coming off this alcohol group here and off this alcohol group here. So we're attaching these phosphate groups via phosphoester links to the alcohol groups that come off the 4th and the 5th carbons of that inositol ring to create phosphatidyl inositol 4,5-bisphosphate, PIP2. Okay, right, now you might be asking, well, why is that not called phosphatidyl inositol 3,4-bisphosphate? Why is that not the 3rd carbon and that not the 4th carbon? What is the difference between this carbon here and this carbon here? Well, the answer is on my picture, absolutely nothing. But in reality, basically, it's a subtle reason. There is a subtle difference between the 3rd carbon and the 5th carbon of the inositol ring. Now, why is there a subtle difference? Basically, it has to do with the optical isomerism of the molecule. Okay, so if you think about cyclohexane 1, 2, 3, 4, 5, 6 hexol, you will realize that that molecule is going to have multiple optical isomers. Now, there are actually 9 different optical isomers of inositol. Okay, there's only one thankfully, that's actually used in biology. Okay, and that's called myo-inositol. And basically, the third carbon of myo-inositol, which is this one here in this phosphatidyl inositol molecule, okay, is subtly different from the fifth carbon of myo-inositol. Okay, so these two are not actually equivalent, basically, and the phosphate group in phosphatidyl inositol 4,5-bisphosphate does come off the fifth carbon rather than the third carbon of the myo-inositol. Okay, so just know that there is a very subtle reason for why it's called phosphatidyl inositol 4,5-bisphosphate rather than phosphatidyl inositol 3,4-bisphosphate, and that there is a difference between phosphatidyl inositol 3,4-bisphosphate and phosphatidyl inositol 4,5-bisphosphate, even though it is subtle and certainly can't be shown on my crude little picture here. Okay, right. So that's the phosphatidyl inositol 4,5-bisphosphate molecule. As I say, it's a normal component of the phospholipid bilayer. It is a phospholipid. Okay, it's not the most common phospholipid to find in the plasma membrane, but it is present in a small amount. Okay, right. So type 1 phosphoinositide-free kinases then. They are going to phosphorylate phosphatidyl inositol 4,5-bisphosphate molecules that are in the plasma membrane on the third carbon of the inositol ring. Okay, so they're going to add a phosphate group onto the alcohol group that comes off this third carbon of the inositol ring here in phosphatidyl inositol 4,5-bisphosphate. And therefore what they're going to turn this into is, if I draw now the product, so once again let's just draw out quickly the phosphatidic acid structure here. Okay, so here, just as a revision, here are the long chain carboxylic acids. Here is the glycerol molecule. Here is the phosphate group, so that's the phosphatidic acid. And then we're going to have the inositol ring here to create phosphatidyl inositol. And then what we're going to now have is phosphate groups coming off the alcohol group, so the third, the fourth, and the fifth carbons. Okay, like so. So one here, and one here, like so. And then we'll colour them in. So this is what type 1 phosphoinositide-free kinase enzymes are going to do to phosphatidyl inositol 4,5-bisphosphate. They're going to convert it into this molecule here. So what is this molecule called? Well, for short, it's just called PIP3 for phosphatidyl inositol, and then you've got 3 phosphate groups coming off the phosphatidyl inositol. To be slightly more correct in your abbreviation, you would abbreviate it down to PI, phosphatidyl inositol 3,4,5, and then P3 to tell people where the phosphate groups are coming off. But again, just like the fact that you can get away with abbreviating PI45P2 down to PIP2, you can get away with abbreviating PI345P3 down to PIP3. Okay, but its full name is, of course, phosphatidyl inositol, okay, the PI. And now we've got 3 phosphate groups coming off the inositol ring of the phosphatidyl inositol. So it's phosphatidyl inositol 3,4,5, and then trisphosphate. Okay, or PIP3 just for short. Right, so that is what type 1 phosphoinositide-free kinase enzymes do. They convert phosphatidyl inositol 4,5-bisphosphate into phosphatidyl inositol 3,4,5 trisphosphate. Now this one, you usually do not have any phosphatidyl inositol 3,4,5 trisphosphate in the plasma membrane. This is not a normal component of the phospholipid bilayer. Okay, when this appears, it has a signaling role basically, and the reason that you don't usually have any phosphatidyl inositol 3,4,5 trisphosphate in the plasma membrane is that there is an enzyme in the, underneath the plasma membrane whose job it is to return the phosphatidyl inositol 3,4,5 trisphosphate back into phosphatidyl inositol 4,5-bisphosphate. So there is an enzyme that is active all the time and which converts PIP3 back into PIP2, and this enzyme is known as P10, and that stands for phosphatase and tensin homolog. Okay, so the P is for phosphatase, the T is for and tensin homolog. Okay, and basically this is an enzyme which sits underneath the plasma membrane and which will go around and cleave off that phosphate group off the alcohol group of the third carbon of the inositol ring and return the phosphatidyl inositol 3,4,5 trisphosphate back down to phosphatidyl inositol 4,5 bisphosphate. So this enzyme, I'll stress it again, is always active, and it's always on the lookout for any of these molecules in the plasma membrane, and if it finds any of them, it will destroy them basically. It'll return them back down to this other one, and that's why usually you do not have any PIP3 molecules in the plasma membrane. It's not a normal component of the phospholipid bilayer. Okay, right, so it only appears transiently basically when you activate phosphoinositide 3 kinases of the first type. Okay, I'm going to probably end up dropping the type 1 phosphoinositide 3 kinases. If I, from now on, say phosphoinositide 3 kinase, I mean type 1 phosphoinositide 3 kinase. We're not going to discuss type 2 or type 3 at all in this video. Not much is known about them, and they don't do the same thing, basically. They do not convert PIP2 into PIP3. They phosphorylate other phosphoinositides, basically. Okay, right. So, what we now want to see... Oh, well, I was on the point of telling you about how it's a transient signal. Okay, so when you activate phosphoinositide 3 kinases, they are going to start converting PIP2 into PIP3, and you're going to get this transient appearance of PIP3 in the plasma membrane, and it causes signaling events, basically. It activates downstream proteins, as we'll see later on, okay? Right, and that's how PI3 kinase achieves its signaling function. Okay, right, so now what we want to do is discuss the structure of phosphoinositide 3 kinase enzymes. We also want to discuss how many different types of type 1 phosphoinositide 3 kinase enzymes there are. Okay, so I've said that there are these 3 different types of phosphoinositide 3 kinase enzymes, type 1, type 2, type 3, but under the subheading of type 1 phosphoinositide 3 kinases, there are loads of different types of type 1 phosphoinositide 3 kinases. Okay, so all of the enzymes we're about to look at are type 1 phosphoinositide 3 kinases. They do this. They convert PIP2 into PIP3, but there are multiple of them basically. There's not just one type 1 phosphoinositide 3 kinase, I'm afraid. Okay, so that's what we're going to now look into. So, all type 1 phosphoinositide 3 kinase enzymes, so I might just put this here. So all type 1 PI3 kinase enzymes are heterodimers, basically, okay? They are composed of 2 separate subunits. Okay, I'll draw this like so. Okay, so there are 2 separate subunits here that make up the type 1 phosphoinositide 3 kinase. Now, one of these subunits is what's known as the catalytic subunit of the type 1 phosphoinositide 3 kinase. Okay, and this is the one that is actually capable of converting PIP2 into PIP3. Okay, so it contains the catalytic domain, which has the active site which can convert PIP2 into PIP3, the major reaction. Okay. The other subunit of the phosphoinositide 3 kinase, type 1 here, which I'll colour in turquoise here, this is what's known as the regulatory subunit of the type 1 phosphoinositide 3 kinase, and its role is to regulate the function of the catalytic subunit. So often it ends up inhibiting the catalytic subunit and stops it from having effect. Okay, and then only when you get conformational changes in the regulatory subunit can then the catalytic subunit become active and phosphorylate PIP2 into PIP3. Okay, right. So what we now want to discuss is how many different types of catalytic subunits are there for type 1 PI3 kinases. Okay, so it turns out that there are 4 different catalytic subunits of type 1 PI3 kinase, okay, and all of these proteins are 110 kilodaltons in size, okay, and therefore they are called P110 proteins, okay, so the catalytic subunits of type 1 PI3 kinase, often instead of calling them the catalytic subunits, people will just call them P110 because they are a protein of 110 kilodaltons. Okay, right, So, there are 4 different P110 proteins then, and these are called P110 alpha, P110 beta, P110 gamma, and P110 delta. Okay, so if you want to build a type 1 PI3 kinase, you have 4 choices of catalytic subunits to use here. Okay, now let me tell you about the genes that code for these. Okay, because the nomenclature for the genes is slightly confusing. Okay, so the gene for P110 alpha is called PI3K. Okay, so for phosphatidic kinase, and then for some reason that's beyond me, they put the 3 after the K, and I find that incredibly confusing. Okay, so the genes, you do not call them PI3K, you call them PIK3, and then because it's the catalytic subunit, you put a C there, and then because it's the catalytic subunit alpha, you put an A there. So the gene for P110 alpha protein is called PIK3CA. Now the first time I saw that, I just thought someone had made a typo, okay, but it was meant to be PI3KCA. Okay, but it's not. It is correctly called PIK3CA. Okay, so that's the gene that codes for P110 alpha proteins. Okay, and the P110 beta and delta and gamma also have genes that are named in a similar way. Okay, so the beta gene is called PIK3, catalytic subunit, and then of course you put a B. Okay, so that's its gene. For gamma, it's PIK3 again, and then catalytic subunit, and then it's G for gamma. Okay, and then finally, the gene for P110 delta is called PIK phosphatidic kinase 3 catalytic subunit, and then of course D here. Okay, so that's the naming of the genes. So the proteins are called P110 alpha, beta, gamma, delta. The genes are called PIK3C, and then it's A, B, G, or D. Okay, right, so that's the nomenclature for these catalytic subunit proteins and their associated genes that actually code for them. Okay, now, the next bit of nomenclature that I want to introduce you to is that the entire type 1 PI3 kinase heterodimer, and I should have put that word somewhere. This is a heterodimer. Hetero means different. Dimer means two-membered subunit, well, two-membered structure. Okay, so because the two structures that make it up are different, then it's a heterodimer. Okay, now, the entire type 1 PI3 kinase heterodimer is named after which catalytic subunit you have here. Okay, so when you are making a type 1 PI3 kinase, we've discussed that there are 4 options available for which catalytic subunit you want to use. We haven't talked about the regulatory subunit. That's going to make it even more complicated. We're going to come to that. Okay. Basically, the type 1 PI3 kinase that you then make is named after which catalytic subunit you chose to put in there. Okay, so if you make a type 1 PI3 kinase where you have used the P110 alpha catalytic subunit, then your PI3 kinase is going to be called a PI3 kinase alpha. Okay, so you'd call it a PI3 kinase alpha. If you use the P110 beta, it will be called PI3 kinase beta. If you use P110 gamma, PI3 kinase gamma, and then of course P110 delta, you would call your PI3 kinase delta. Okay, right, so that's a bit of nomenclature there. The next thing I need to say is that type 1 PI3 kinases are split up into 2 further classes. Okay, so there are the type 1A PI3 kinases, and there are the type 1B PI3 kinases. Okay, now the type 1A PI3 kinases are PI3 kinase alpha, beta, and delta. Okay, these are the ones which can be activated by receptor tyrosine kinases. Okay, the type 1B PI3 kinase is then PI3 kinase gamma, and this is the one which cannot be activated by receptor tyrosine kinases, as we'll discuss. Okay, so remember this naming here just refers to which catalytic subunit you have used. So if you use P110 alpha, P110 beta, P110 delta, you end up with a PI3 kinase alpha, beta, or delta, and that's called a type 1A PI3 kinase. Okay, if you use P110 gamma, you end up with a PI3 kinase gamma, and that's called a type 1b PI3 kinase. And what's key here is that the difference between them is firstly that they're going to be activated by different things, okay? So the receptor tyrosine kinases are going to be able to activate the type 1As, but not the type 1Bs. And the reason that receptor tyrosine kinases can activate these ones and not these ones is that the type 1As all have certain types of regulatory subunits. So all of these types of PI3 kinase, where you've used P110 alpha, beta, and delta as your catalytic subunit, they're going to have a certain type of regulatory subunit that can be used and attached to them, whereas PI3 kinase gamma, where you have used P110 gamma as your catalytic subunit, that's going to have totally different regulatory subunits that can be attached to it, okay? And we'll have a look at the regulatory subunits in the next video.